Hi, my name is Onnis from the University of Siena, and I will describe the main findings of my GEM paper entitled SARS-CoV-2 Spike protein suppresses CTL-mediated killing by inhibiting immune synapse assembly. During viral infections, naive CD8 T cells are activated to CTLs and migrate to the site of infection, where they form specialized signaling junctions known as immune synapses with viral infected cells to promote their apoptotic demise. We hypothesized that in severe COVID-19 patients, SARS-CoV-2 may modulate CTL function by targeting immune synapse assembly to escape CTL-mediated killing. To infect away epithelia, SARS-CoV-2 binds to the ACE2 receptor through the envelope glycoprotein spike. Whether ACE2 is expressed in CTLs is currently unknown. Interestingly, we found that ACE2 is undetectable in freshly purified CD8 T cells from human healthy donors, but is upregulated during the differentiation to CTLs. We next investigated the outcome of spike binding to ACE2 in CTLs in immune synapse, and we found that pretreatment of CTLs with spike suppresses immune synapse formation as assessed by defective synaptic accumulation of the T-cell receptor and the tyrosine phosphoproteins in the presence of spike. Spike-pretreated CTLs also showed defect in centrosome polarisation toward the synapse. Additionally, the convergence of lytic granules around the centrosome was impaired in CTLs, thereby affecting the delivery of lytic granules at the synaptic area, which is important for their killing capability. To investigate if the synapse defect in the presence of spike affects the functionality of CTLs, we carry out cytotoxicity assays by incubating spike-pretreated CTLs with target cells. Overall, spike suppressed the ability of CTLs to kill target cells and to produce cytokines. At this point, we ask whether the effect of spike on CTLs are mediated by ACE2. We found that ACE2 accumulates at the synaptic area. However, the localization to the synapse was impaired in the presence of spike, suggesting that spike binding to ACE2 may modulate immune synapse assembly by preventing its correct localization. Finally, we found that CTLs from bronchoalveolar lavage but not peripheral blood CTLs from COVID-19 patients display severe defect in immune synapse signaling, suggesting that in vivo CTL exposure to spike in the respiratory tract during acute infections compromises their ability to form functional immune synapses. Collectively, we report a new mechanism of immune evasion by SARS-CoV-2 based on direct disassembling of CTLs to form immune synapses through spike protein binding to ACE2. This mechanism could contribute to explain the failure of the immune system to control SARS-CoV-2 infection.